Morning, everybody.
Morning.
Yay, Thursday. It's almost Friday, which means it's almost the weekend. Okay, so we have a ton of stuff to do today. Yeah. Well, it's not going to be that long before I actually finish this course. It's kind of crazy. I think it's going to be, there's an exam next week, and then there's like another week, and that's kind of it. Yeah, it's pretty wild taking genetics in the summer. That's for sure. All right, so okay, so what we've got to do today is have a quick look at transposons, which are pretty funky things, and actually one of the reasons why we, oh God, my dogs, um, we have a lot of genetic cruft, I guess, but also how we've gone attain new functions too, which is pretty interesting. So we're going to go through that, and then we're going to start on gene expression, right? So hopefully today we'll get through most of the overview and transcription. That's the plan. So with no further ado, we're going to start on transposons. And transposons are actually kind of a little bit close to my own heart because I did a bunch of work with these in Australia and New Zealand for one of my postdocs, and they're pretty cool. And so transposable elements are, let's move this back down there, essentially mobile genetic elements, right? So they are independent pieces of DNA that can move around in a genome, right? And actually a little bit of history, which is always fun to talk about. These were discovered in the 60s, I think, by a researcher called Barbara McClintock. And she was working on the genetics of maize or corn, or the stuff with the kernels. And she noticed that there was this weird kind of non-Mendelian spotting pattern, right? And she couldn't figure out what it was caused by, right? Because the crosses didn't work out. And so she basically found this, whatever was causing the phenotypic change, right? It's basically something was knocking out a pigmentation gene, right? Similar to what we looked at in the corn genetics lab. And essentially what it was a genetic element moving into that gene and knocking it out, but it did so randomly, right? And so there was no kind of mappable inheritance patterns because it did it on a like literally a cell by cell basis. And she was widely derided for this theory, right? She was kind of told that she didn't know what she was doing. And it turns out she was right. And she got a Nobel Prize for that, which was pretty cool. And so transposable elements they're pretty cool. I like talking about them. They're just something kind of almost alien, right? You could almost think of them as being genetic parasites, right? Which just kind of move around in your genome to their own ends, right? Not necessarily to benefit us. And they make up a huge amount of our genome, right? Up to 40%, 45%, right? So there's a large part of our genome which doesn't code for genes is made up of transposons. And that causes all kinds of funkiness, right? And so actually we'll get to this later once we go through the mechanism, but it potentially can result in all kinds of mass chromosomal movements and also gains of function in terms of new traits, right? So it's actually potentially had a big role in our evolution, which is also pretty cool. So there are lots of different types. We're going to cover the main types today, but I just want to go through the basic mechanism. Right, and so the idea with transposons is they have a recognition site. right? So, for example, I'll give you the one that I worked on. It's called Piggyback. Don't know why. It recognizes TTAA, right? So anywhere there is a TTAA in your genome, Piggyback could jump into it, right? So how does it do that? So actually, I'm going to jump forward a little bit. Need to get a better image. All right, so essentially a transposal element consists of a piece of DNA, could be several kilobases long, and it has what's called a terminal inverted repeat, right? So this part here, green and yellow, is the transposon. In the middle is something called, is a gene which encodes a protein called transposase. So transposase is the protein that recognizes the repeat. I'll get to that in a second. Good question, though. You have to forgive my spelling. It cuts the DNA and inserts the transposon. So transposons carry the mechanism or the means by which they move themselves. Now the inverted repeats are what the transposase recognizes in the transposon itself. And they're inverted because it's kind of difficult to explain why, but essentially it's like they're bookends. So if you have the same sequence but in kind of mirror image at the end of something, that's often what's recognized as being this is the either end of this piece of DNA, right? And so the transposase itself recognizes that, and that's what it uses to cut the transposon out and stick it somewhere else, right? So again, transposon, terminal inverted repeats on either side. And then in between is typically the coding region for one gene, and that is a transposase. That's what's responsible for cutting out the transposon and sticking it somewhere else. And so this is a kind of a simplification in a sense, right? But what happens is the transposase recognizes its target, and then it cuts the DNA, so it makes a double-stranded break. However, it's not a butt-ended double-stranded break. That's really, really important. It's what's called a staggered cut. So in one strand, it's cut over here, and the other strand, it's cut over here, so that this bit of DNA overlaps, right? Does that make sense? That's kind of like the thing behind a staggered cut. So what you end up with is what are called sticky ends, right? These are overhanging single-stranded bits of DNA. Now, when you have that overhanging bits of DNA, transposon gets inserted in the middle, right? And so what that leaves is these single-stranded bits of DNA on either side, and those get filled back in by DNA polymerase. So what that does is if you see the center part of this cut reads TCGAT. right? Now, because the transposon, yeah, sure. Here we go. So, no worries. The staggered cut leaves this TCGAT as an overhang, right? So that's the sticky end bit. So when DNA polymerase fills that in, what you end up with is two copies of that TCGAT, right? The bit that was the overhang, right? And so these are called flanking direct repeats. And that's essentially the footprint of a transposon. If you wanted to find transposons, right, you could either look for the, zoom out a little bit, terminal inverted repeat, which essentially is the same sequence, but kind of going in as a mirror image, essentially. So as you can see, ATC, GCA at the top here, ATC, GCA at the bottom here, going in the other direction. But also you get these flanking direct repeats. So even when a transposon jumps back out again, right? So depending on the transposon, these are mobile, right? They move around on their own accord. If this transposon then jumped out again, go somewhere else, it would leave behind a doublet, right? These two flanking direct repeats would be stuck together and you'd have a footprint. So that's called an excision footprint. So obviously having a transposon jump into a coding region is bad news, right? Because you're going to have a large, many thousand base pair chunk of DNA in the middle of a gene, right? Which is generally not good. But even when that jumps back out again, almost certainly it's going to leave behind, well, it will leave behind a flanking direct repeat, which will still disrupt gene function if, say, it's in a coding region. So transposons are incredibly mutagenic, not just from them going into a piece of DNA, but also when they leave, they'll leave behind their footprint, which also will disrupt at least a coding region. Okay, does anybody have any questions about the mechanism? So it involves cutting the DNA, double-stranded break, by the transposase. Transposase, depending on the mechanism, cuts the transposable element out of where it's at, inserts it into the DNA, and basically kind of ligates it in, as would DNA ligase, right? So it's both a endonuclease and a ligase, which is pretty cool. And then the host, or the kind of cell's DNA polymerase, fills in those gaps that are left by those overhangs, generating these flanking direct repeats. All right, so here's a question. What do you think is going to happen if a random mutation, just through, I don't know, whatever cause, inactivates the transposase, right? So it generates a null mutation in the transposase that transposon needs to move around. What's going to happen? Yeah, well, I mean, the mutation will be incorporated, let's say, during DNA replication. But what would the consequence on the transposon be? It would cause an error. So that if the transposase doesn't work, what's going to happen to the transposon? There's nothing to be filled. It's not going to move, right? And so if you look at the human genome, you'll find a large number of dead transposons. So these are transposons where the transposase in between is inactivated. And so that transposon can no longer move because there's no transposase to move it around. And so it just dies, essentially. And so a lot of our DNA, our genome is filled with not just transposons, but also dead transposons, which over time have accumulated mutations so that transposase no longer works. Or if there's a mutation which occurs in the terminal inverted repeat, or one of them, that won't be recognized by the transposase, and so again, the transposon stays put. So they're pretty interesting also, not just the mechanism, but also from the perspective of evolution as well, like the evolution of our genome. So there are a couple of different types, well, there are lots of different types, but broadly speaking, there are DNA transposons, right, which are just DNA. And there's also DNA transposons that transpose via an RNA intermediate, right? So there's still DNA in the genome, but to transpose into a different site, they get turned into RNA. So these are called retrotransposons because they have to get turned back from RNA into DNA. So DNA transposons can either transpose via non-replicative transposition, right? So that's cut and paste, take one out, stick it somewhere else, or replicative transposition, which is called copy and paste, right? So the transposon is copied and stuck somewhere else. And so let's have a look at retrotransposons. So we have the same deal here. We have our flanking direct repeat, which was caused by the previous insertion of that transposon. And we have our transposon in the middle, right? So this is actually transcribed, right, by RNA polymerase into RNA. And then it's reverse transcribed, which must be by a reverse transcriptase, which is present in the transposon itself, because we don't have that as an organism, right? We are not able to do that ourselves. That function has to be supplied kind of exogenously. Much as it is if you think about HIV, for example. So HIV is a retrovirus, and it actually incorporates itself into our genome by turning its RNA into DNA by reverse transcriptase, and it provides that as part of its viral genome. Anyway, so that RNA gets reverse transcribed into double-stranded DNA. And that double-stranded DNA is now what gets inserted into the new site, right? Via the same mechanism that we talked about earlier, generation of staggered cuts, insertion, and the filling in of those staggered cuts to create the flanking direct repeats. So as you can see, depending on how active this transposon is, you can actually generate an awful lot of copies of this transposon in the host DNA fairly quickly over kind of evolutionary time. So I'm not sure, do I have it? Now that's another way of thinking about this is can be either cut and paste. That's when the transposon is cut out and inserted somewhere else. Or copy and paste. So just think about writing stuff in Word, I guess. Right, it's kind of the equivalent. Control C versus Control X. Where the transposon is copied and the copy is inserted somewhere else. And that somewhere else is basically wherever oh, come on, can't see what I'm typing out here. So it's basically wherever there's one of those recognition sites. And if you think about Piggyback, it has a 4-letter recognition site, TTAA. And so if you think about how often that would occur, that would be 4 to the power of 4. What was 16? I can't do the math on that. Whatever 4 cubed is. Still, a fairly small number compared to 6 billion base pairs, which we have in our genome. So these things can move around a lot. And whenever they move, they're going to generate a mutation, right? So they are highly mutagenic, right? And it also depends on how much they move around. If they move around a lot, they're going to cause more mutations. If they move around less, then they'll cause fewer ones, right? And so the cool thing is, though, is that obviously mutation is typically bad, right? So if one of these transposons transposes into a coding region or into a regulatory region, they can disrupt it and cause a loss or reduction of function mutation, typically a loss of function, right, where something just doesn't work anymore. However, these transposons, the transposase is a DNA binding protein. And actually from doing some reading about these a little while back, there's a quite good evidence that transposons moving into regulatory regions, and we'll get to what those are in a little bit, has actually added function. So they essentially provide DNA binding sites for other proteins which may recognize them and alter the transcription from that locus, right? So even though transposons are often thought of as generally a bad thing, there's quite compelling evidence that they've also driven change in our genome, which has been selected for, right? And that's part of how, you know, we became who we are. So they're pretty funky things. And also what's even neater is that they can actually result in chromosomal changes, right? And so essentially, the outcome is that these transposons, because they're identical, if you have identical bits of DNA in different chromosomes, those different bits of DNA can actually serve as sites of homologous recombination. And so if you think about like chromosome 1 and chromosome 2, right, these shouldn't stick together because they're very different. But now we actually have bits of DNA that match on here and here. And so potentially you can get fusing of chromosomes, you can get bits of chromosomes flipping over, all kinds of chromosomal funkiness, basically, which again, generally speaking, is going to be bad, right? Because, you know, we like having our chromosomes the way they are, but potentially can lead to kind of novel traits that are advantageous, right, and can be selected for over time. And so it's thought that there's at least one, I think, between chimpanzees and humans, where there was a chromosomal fusion between our last common ancestor and the chimpanzees that didn't happen between our last common ancestor and us, however many millions of years ago that was. So it's actually pretty neat. So part of how we are potentially isn't just about what genes we have and how we turn those on and off, but also what transposons have been jumping around like fleas in our genome over the last several million years. So it's kind of neat to think about. Anyway, something a little close to my own heart, given that I've spent a couple of years working on these things. Does anybody have any questions about transposons before we move on to gene expression? Bit of a quick run through, but it's something that's cool to talk about. And it's also something that is very much non-Mendelian, right? So none of this follows the laws of Mendelian inheritance, right? So because these DNA elements are independent and mobile, how they move around is going to differ from individual to individual, from cell to cell as well, right? Not just from me to my children, but different cells within my body are most likely going to have transposons in different places, right? And so this is a very non-Mendelian inheritance. It's actually, they're just very weird things to talk about. Essentially, this idea that we have bits of our DNA that, I don't know, essentially, I mean, it's hard to think of them as anything other than parasites, right, which exist in our genome. Sometimes they're good for us, but most times they're not, and other times they don't make any difference at all. So it's a cool thing to talk about. But again, the main points of interest are really, how does it work? This being the mechanism of transposition, right? So the staggered cut, insertion of the transposon, and then the filling in of the gaps to create the flanking direct repeats. What a transposon looks like. So it has these terminal inverted repeats which flank a transposase gene. How retrotransposons work, which is via this RNA intermediate. And then really the consequences of transposition. Mutation, sometimes that mutation doesn't make any difference. Sometimes it really does, depending on where it inserts. And big consequences in terms of how transposons and transposition can change the chromosome structure of an organism. So those are the main points to take away from this. Cool. Okay. So if no one has any questions, I'm going to move on to gene expression. Okay, so this is going to be a kind of a big chunk of the rest of the section, right? So basically gene expression is the rest of this exam section, right? So this is actually a fairly short section, although we've got a lot of information to cover in this section. And so really it's going to be genome structure, transposons, transcription, RNA processing, and translation. Those are the topics that will be on exam 3, which will be next Thursday, right? So this is a real fast run through. And then the rest is going to be about gene regulation and a little bit of biotechnology and transgenics, depending on time at the end. Right, so we're really what we're starting today is kind of the bulk of what will be on exam 3. And so there is a lot to take in for this. There's a lot of details. There's a lot of like things to remember. Best way to review this, Galen? That's a good question. So everybody's different, right? And so whatever tip I give you might not necessarily be the best one for you. But the way I would go about this is to try and kind of work. Actually, I'd draw it out. So, you know, without using your notes, right? Because this is a good way of testing recall, and then you can figure out what you don't know, right? That's really the key here. Try and draw this out, right? Can you draw out all the steps? You know, if you have like a big poster-sized piece of paper, can you draw out all the steps in gene expression, say in bacteria, let's say, which is somewhat simple? You know, can you go through all of transcription and transcription termination, and then go through translation and generation of a functional protein? Now, if you do that, and you'll probably, you know, find bits, let's say the mechanism of translation, or, you know, row-dependent transcription termination, right? You might remember intrinsic, but you can't remember extrinsic. That will show you what you don't know, so then you can go back and figure out how that works. So really, one thing that I stress in terms of studying is you have to study actively. Just looking through your notes and looking through the PowerPoints isn't a bad thing. Oh, Erica, send me an email after class, Erica. We'll figure this out. So yeah, don't worry about it right now. Send me an email and we'll sort it out. It's not a big deal. So yeah, you got to figure out what you don't know, and you're never going to figure that out by simply just looking at what you do know or looking at the notes that you've taken. The key is to figure out what you don't know. And the best way of doing that is see if you can work through this unassisted, unaided on your own, right? Without access to your notes, even if you have your notes available in the exam, you don't want to have to rely on them. And so when you do figure out what you don't know, then you can kind of drill down into that with whatever resources that you want to use. It could be this stuff that I'm giving you. Maybe if it didn't work the first time, that's not the best material. You might want to look it up on, you know, Wikipedia or Khan Academy or any of the other online resources. You know, maybe explore it a little bit further, check the textbook if you have one, or other sources of information, and basically teach yourself this stuff. And so that's what I do whenever people ask me a question, or, you know, kind of reading through something like, you know, I'm not really sure that makes complete sense to me. And I'll go and spend sometimes like an hour, you know, tooling around on the internet, checking stuff out, figuring it out in my own head, and then I'm good. Then I understand enough, and then I can, you know, do a better job of teaching it to you. So that's what works for me. right? To be completely honest, I never read textbooks because they just send me to sleep. It's just not how I can access information, right? Yeah, I mean, they're good source of information. There's no doubt about that. And if you use them as a resource to go and find bits, then they can work very well. But I can't just sit down and read a textbook. It just doesn't work for me. Different for other people though. So what works for me is kind of, jumping around, how does this work? Oh, okay. And what about this bit? And checking out from there and just kind of figuring out in my own head. So I'm pretty good at figuring out what I don't know, partly because, you lot will ask me questions and I don't know the answer. So there you go. That's one thing. Um, Also, I recognize when I don't understand stuff. Yeah. So no cards are good, but you don't want it to be copying out stuff that you've already written. So that's kind of one of the key points I'm trying to make is that this has to be active learning, right? So copying out notes on note cards, again, reinforces what you already know, but it doesn't necessarily help you figure out what you don't know. And obviously, those are the things that really matter because that's what's going to trip you up in an exam. And so, really, the way to find out what you don't know is either to, pull your notes down, close your computer, and just go, okay, gene expression, how does it all work? What are the different steps? What are the different things? How does it differ between eukaryotes and prokaryotes, right? What's the mechanism of transcription, termination, and bacteria? Well, there's two. Can I just draw them out? And if you go, oh, um, can't. Ta-da! Now you know something that you don't know, right? And that will then make you go and look for it. And it's actually the process of looking for something, right? Actively seeking out information. That's what helps it stick in your head, right? Because you're basically making new neural circuits, right, to contain that information and that understanding, right? Rather than simply reinforcing neural circuits that already exist. It's a bit of an oversimplification, but that's a general idea. Another way is to have someone else quiz you, right? Or to use the question sets at the end of each chapter in the textbook. That's actually, in my opinion, the most valuable thing in the textbook is the question sets, right? Some of which I think, it's a long time ago now, I might have actually used to make exam questions. I can't honestly remember, to be honest, right? And so the key is always, how do you figure out what you don't know, right? And you can't do that by looking at notes or PowerPoints or watching me chunter on the screen or whatever, because again, it'll feel like you know it until it comes to the exam and they're like, oh shit, I don't know that, right? I thought I did, but clearly I don't. You want to figure that stuff out ahead of time. So that's how I would suggest studying. It takes a lot more effort. And sometimes as well, it feels like you're not making as much progress. Like if you want to get through all of Blur in a certain amount of time, it can take you a lot longer to figure all these things out, and teach them to yourself, essentially. But it's much, much, much more effective. Orders of magnitude. Yeah, it just takes a lot of time. And, to be honest, I typically spend about an hour before, like every morning before teaching Tuesdays and Thursdays, just going through the PowerPoints, looking stuff up online, kind of going through my head, some of this stuff I know without even looking at it, other stuff I need to refresh myself on. So even it's not just me sitting on my couch with my dog next to me, kind of running through stuff, even I need to go through and study, right, to teach this. Because there's a lot going on, right? There's a lot of details, there's a lot of stuff to hold in your head. And also I need to be able to understand it several layers better in order to be able to teach it, right? That's kind of one of the difficult parts of teaching. So yeah, give all that a shot, and I'm pretty sure you'll find that you're more successful when it comes to exams, right? And again, some people don't study like that at all well. They just don't. They have different means, and that's totally fine, right? Whatever works best for you. But if you're hunting around for something different to try, I'd suggest trying that. But also going over stuff, you know, over time, right? So don't leave revision until the last minute because, you know, it's just too much stuff to take in. You can benefit from building up those neural circuits, essentially. Yeah. I mean, in a sense, same for Gen Bio 1 was actually a big part of this too when I taught it, but genetics too it's really about learning how to study as well as learning genetics, right? The two things are not really separable. And so one thing I will tell all of you, like ahead of time, like full disclosure, exams 3 and 4 are harder for most people than exams 1 and 2. The reason they're harder is because a lot of the math that we did in 1 and 2, if you got that, you were pretty good for a chunk of the material, right? Yeah, I know, not for everybody, right? Some people actually do a lot better the last half of the course because they're not very mathematically inclined, right? But there's a lot of details, a lot of how does this all work together? Why is this different from this? Why did this result in this outcome and this result in this outcome, right? There's a lot of very abstract, kind of intangible stuff. And a lot of people really struggle with that, right? So me, I'm a molecular biologist, right? So this stuff just comes naturally to me, right? Most people aren't. Most people are like kind of more fluffy panda biologists in their inclinations. Not that there's anything wrong with that. It's just kind of generally speaking, you're either like big organismal or molecular, right? And we're now very definitely moving into the molecular side of things. Yeah, don't get me started on pandas. I think I'm developing somewhat of a reputation amongst A&M students for my just absolute, I'm not sure hatred's quite the right word, but strong disdain for giant pandas. But anyway, we don't have time to get into that. That's something else entirely. Okay, so yeah, I know. Yeah, yeah, and they're pretty evolutionarily useless, you know, in my opinion. But I'm not going to get into that. Yeah, totally useless. Anyway, so in terms of gene expression, what we're talking about is the whole deal from DNA to protein. Typically speaking, the things that do things are proteins. Not always. There are RNAs that do a bunch of stuff, important things. But when we talk about gene expression, we're typically talking about the process of taking DNA or the information in DNA, more importantly, creating an intermediate, which is mRNA, and then translating that intermediate into a functional thing, protein, right? And this picture here is really what it looks like in eukaryotes. And you know it's a eukaryote because it has a nucleus. Ta-da, simple, right? But what we're going to do is talk about both prokaryotic and eukaryotic gene expression, as we will do when we talk about gene regulation. And so there are many commonalities between the two. but there are also quite a lot of differences as well. Okay, so basically, gene expression is the process by which information in DNA gets turned into molecules that determine the phenotype of an organism. Right, and those molecules are typically proteins, but not always. They can also be RNAs, and so we've actually got some RNAs up here. First two are involved in translation, ribosomal RNA and transfer RNA. We'll talk about those more later. The last three are involved in all kinds of funkiness, right? mRNA you already know of. Small nuclear RNAs and SNO and microRNAs and stuff have all kinds of weird functions, which we'll talk about in terms of gene regulation. So there's, as with all of everything I'm teaching you, there is just so much more kind of to dive into. You could get lost, right? So this is like the Cliff Notes version of gene expression. And it really involves two main processes, at least in both prokaryotes and eukaryotes. Transcription, which is turning DNA into, well, it's not turning, I always use this, like, language is sloppy, right? sRNA, up here. So SN is small nuclear RNA. Those are involved in splicing or RNA processing. They actually make things called small nuclear ribonuclear proteins, otherwise known as SNRPs. SNO, I don't actually know what SNO stands for. There you go, something I don't know. I know that it is a regulatory RNA. MicroRNAs are small RNAs that are involved in gene regulation. There's PIWI RNAs, PIWI. Those are also involved in that. There's small interfering RNAs. There's actually a whole universe of RNAs that don't encode proteins. And I know like that much of them. It's always actually kind of fun telling my students that there's a whole bunch of shit that I don't know. Just goes to show this is all a learning process, right, for everybody. Anyway, so yeah, the R and the T is RNA equals ribosomal RNA, which is a ribozyme, which is a pretty cool word. That's basically an RNA enzyme. tRNA equal transfer. And also obviously protein being the thing that does stuff. All right, so essentially we have DNA. In prokaryotes, that DNA is just in the cell, right? So, but we still need to get the information in that gene into a form where it can be translated, right? So that process is transcription. We need to make RNA that can be used. Essentially, mRNA is an intermediate, right? It doesn't have any other function in and of itself other than to serve as a intermediate between the DNA and the functional protein, right? In eukaryotes, that RNA has to be processed, right? So we'll spend a bit of Tuesday talking about that. And then that mRNA in prokaryotes, that's straight from transcription. In eukaryotes, it's after RNA processing. That gets translated to make a polypeptide, which ends up being folded into a functional protein. And obviously, there's many more steps after that to generate a functional protein. Okay, so all of this so far should be fairly straightforward, kind of building on stuff that you've already covered in previous courses, right? So now what I'm going to do is talk a little bit about kind of gene structure, which is very different between prokaryotes and eukaryotes, and a little bit about kind of directionality, right? So I talked a lot about that in terms of directionality of DNA, right, which is really important in DNA replication. It's also super important in transcription too. And so the direction of DNA is essentially transcribed into the direction of the mRNA, right? So mRNA is only made in the 5 to 3' direction. You can only add new nucleotides on the 3' end, just as with DNA. So another point to, and we'll get kind of jump ahead a little bit and then get back, is that DNA, 2 strands, right? So I'm going to type this out because it's important. One serves as the coding strand, right? So that contains the info for the protein, essentially, right? The other strand is the template strand. This is used to synthesize. Bollocks. mRNA. You know what I mean. So this is actually a really kind of funky thing to think about. Actually, let's draw this out. Probably be easier to get a little bit of a different setup. Woohoo! That's going to work pretty well. All right, got new couch covers, so slightly different setup for the whiteboard. So if you have that's really not a very straight line. Never mind. Right. So this would be your DNA double helix. Now you can only synthesize mRNA in a 5 to 3' direction. Let me just get this off to make this bigger. Right? So if this had our, how do I want to put this? Right, so this has our info, our gene essentially, right? The information that we need to turn into protein at some later point. Can we synthesize our mRNA using this strand? Right, so don't forget, our info has directionality. So we want to make an mRNA that goes in that direction too. But mRNA has to be made like DNA in a 5' to 3' direction. So if we pull this open, imagine these little dashes are the bases. You just have to use your own imagination. You can even put letters and stuff in there if you want. Which strand am I going to have to use for the template? Can I use this strand? Or would I have to use this strand? I'm going to take your answer, Galen, when I was pointing at this one. So let me write it out and it will become a lot more obvious. So if we want to make an mRNA strand that goes in the same direction as the information in the DNA, we're going to have to use the other strand as a template. So this is called the coding strand. Because that's what contains the information. This is called the template strand. Because this is what's used as the template to make the mRNA molecule. Now, turn around. If the information is on the bottom strand, going that way, the top strand is used as the template, because that's the way in which the mRNA strand can be synthesized in the same direction as the information. Also, if we do actually put in some letters, this is going to be TAGC. What we're going to get is, this is going to be AUCG. Now, let me actually match those up better. That was a sloppy job. So the only way that you're going to get the same letters, same information, is if you use the other strand, the complementary strand, to synthesize the mRNA. So now, because T is here, you're going to get an A, which is the same as the coding strand. Here's an A, so you get a U, which is the equivalent of thymine in the coding strand. If it's C here, you'll get a G in the template strand, which means you'll get a C in the mRNA, which is the same as the coding strand, and so on. So both strands are important. Hello again, Robert. We've got the strand which contains the information. We've got the strand which is used as a template. Oh, I think my wife's having toast for lunch. That smells really nice. Starting to get hungry. Oh, shit. It's actually end of, there you go, end of the lecture. Woohoo! All right. Sorry, I was getting kind of a little lost in time. But one thing that I want to end up with is you have two strands. If the information is going that way on that strand, the other strand's used as a template. This strand here, if there's information kind of down here, can be the coding strand too, right? It's just the gene is going to go in the other direction. So it could look very much like that. Coding strands on the bottom, going from right to left, templates strands at the top. It simply depends on which strand contains the information that's going to get turned into mRNA and then going to get turned into protein. It's kind of neat actually having this little whiteboard. Cool. All right. That's actually taken us to time. So I'm going to leave there and we'll pick up again in 15 minutes. So go stretch your legs, grab yourself some tea or something, and I'll see you in a little bit.